In this video, we're going to be discussing polymers and the first of our polymeric macromolecules, that's carbohydrates. And by the end of this video, I want you to be able to differentiate between dehydration and hydrolysis reactions in regards to making or breaking down polymers. I want you to be able to identify the monomers of carbohydrate polymers. Name the covalent bond that connects the monomers of carbohydrate polymers. I want you to be able to describe how monosaccharides can differ from one another. I want you to identify a few key disaccharides. And I want you to be able to describe the key differences between structural and storage polysaccharides. So let's take a moment to discuss the definition of a polymer. So here we have a polymer and it's amylose starch. And what you notice is that it is made up of repeating units, and those units are called monomers. In this case, the monomer is the monosaccharide glucose. And so we're just adding glucose over and over and over and over again. That's a polymer. Theoretically, this starch can go on forever, right? We can keep adding glucose to the end, to the end, to the end, to the end. And so we have this very long chain of monomers that can, again, theoretically go on forever. Now, the reactions necessary to build a polymer, to link a monomer to another, to a growing polymer, is called a dehydration reaction. So here is our growing polymer. Here is our unlinked monomer, and what we're going to use is this dehydration reaction to form a covalent bond between the monomer and the growing polymer. And the way that works is we lose an H from the polymer, we lose an OH from the monomer. That comes out as H2O, dehydration, losing water, right? That's what that means. And then we end up with our covalent bond with the new monomer added. So dehydration reactions are the reactions necessary to create polymers, to add monomers over and over and over again. The opposite reaction is the hydrolysis reaction. So if we have a polymer and we want to cut off one of the monomers, we use a hydrolysis reaction. And in this case, we use a molecule of water to break that monomer off. Hydro, water, lysis, to break, hydrolysis. Now, many of the enzymes in your saliva and in your stomach are hydrolytic enzymes. That means those are enzymes that catalyze, that speed up these hydrolysis reactions, the breaking down of polymers. All right, so with that said, what I'd like to do is help you study macromolecules. There are going to be 3 important polymers that you're going to learn in the next few days. And what I want you to do is I want you to memorize what the polymer is, what the monomer is, so what we're adding over and over and over again, and then what's the name of the covalent bond that connects that monomer over and over and over again. So we have 3 polymers: carbohydrates, which we're going to be learning about now. We have proteins and nucleic acids. So let's fill in this table here. The name of the monomer that makes up carbohydrate polymers are monosaccharides. Monosaccharides. And the covalent bond that connects a monosaccharide to a growing carbohydrate chain or polysaccharide chain is a glycosidic bond. All right. Do you know the monomers of proteins? Probably hear them a lot. Amino acids. And the covalent bond that links amino acids together is called a peptide bond. And finally, nucleic acids, right? That's going to be your DNA and your RNA. And the monomer of nucleic acids are called nucleotides. That's your adenine, your guanine, cytosine, thymine, right? Your A's and G's and C's and T's. And the name of the covalent bond that connects nucleotides together is the phosphodiester bond. You should be filling out tables like this constantly, filling them out, remembering them, filling them out again. And this will be a common way that I suggest that you study in several of the different subjects that we're going to be covering. So let's go back to carbohydrates. Now, there are three general classes of carbohydrates that we are going to discuss. Monosaccharides, those are going to be your monomers of disaccharides, putting two monomers together, two monosaccharides together. So here, glucose is a monosaccharide, sucrose is a disaccharide. And then we have polysaccharides. That's our carbohydrate polymer, right, where we have lots and lots of monosaccharides connected to one another in a big, long chain. All right, monosaccharides. Let's talk about monosaccharides in some detail. First, what we see is that it only contains carbons, hydrogens, and oxygens in this ratio. So for example, glucose is C6H12O6. These monosaccharides are major nutrients for the cells. They are also building blocks for the cells. They are oftentimes stored in cells as polysaccharides. And even though most commonly in the cell a monosaccharide looks like this, it also can exist in a chain form. And I want you to focus on the chain form here so that you can look at it in a critical way without having your eyes glaze over. So what a monosaccharide is nothing more than a chain of carbons. Okay, now that chain can vary in length, as we'll see in a moment. And in one of those carbons, exactly one of those carbons is a carbonyl, right, that double bonded oxygen. And all the other carbons have a single hydroxyl group connected to it. And then everything else is filled out with hydrogens. And that's it. You take a carbon chain, add a carbonyl, add one hydroxyl group to the other carbons, and then fill things out with hydrogens. It is a very easy molecule to think about when you see it in those terms. Let's think about the way different monosaccharides, how monosaccharides can differ from one another. And one of the key differences is the nature of that carbonyl. Now that carbonyl group can be an aldehyde. It can exist at the end of the carbon chain. In that case, we call it an aldose. So this is an aldose monosaccharide. Or that carbonyl can exist somewhere in the middle of the chain, and we call that ketose. Okay, that's a ketone group, and so we call this a ketose. Right now, all the other carbons are bounded to a single hydroxyl group and one hydrogen, and filled out with hydrogens. Okay, so first way we can distinguish two monosaccharides: are they an aldose or are they a ketose? Next, the different monosaccharides will differ in the number of carbons in their carbon chain. If it has 3, we call it a triose. If it has 5, we call it a pentose. If it has 6, we call it a hexose, and so on and so forth. Okay, so that's another way that monosaccharides can be different from one another. And finally, they can differ in how the atoms are arranged around the carbons, how the different groups are arranged around the carbon. So if we have this molecule here where it's both 1, 2, 3, 4, 5, 6, so both of these are hexoses. Both of them are aldoses, and yet they're still different molecules because look, around this carbon here, they are different. The hydroxyl group here is on the right side, where here it's on the left side. It might sound like a very subtle difference, but it's enough to give them differing chemical properties and be able to maybe interact with enzymes or other molecules differently. So although it looks like a subtle difference, it plays a big role in the nature of those molecules. The last difference that we may see between monosaccharides is how they form a ring. Now, like I said, most of the time monosaccharides exist like this in solution, in the ring form and not the chain form. This is the reaction. This is how a chain form of a monosaccharide goes into the ring form. And the way it works is one of the hydroxyl groups is going to form a covalent bond with the carbon of the carbonyl. So this oxygen is going to covalently bond to this carbon. And when it does so, we lose this hydrogen, and this carbonyl becomes a new hydroxyl group. So what we get here is, see this blue oxygen? That's this one here. And see this carbon? That's this one here. So now we have a hydroxyl group and a hydrogen there. Okay. Now the way this works is sometimes the molecule can bend this way to attack that carbon, or it can bend the other way to attack that carbon. And depending upon which route it goes to become in the ring form will determine the type of anomer it is. Okay, so for example, if it goes into the chain form in one way, the resulting new hydroxyl group will be on the opposite side of the ring as that C6 carbon, the carbon outside of the ring. And we call that the alpha anomer, or alpha glucose in this particular case. If it happens in such a way where that new hydroxyl group that's formed is on the same side of the ring as that sixth carbon, we call this beta glucose, or the beta anomer of the glucose. So again, this looks like really a very subtle difference of where that hydroxyl group is, either up top here or down below here. And yet it has an extremely important effect on the structure and therefore function of the molecule that they are involved in. So for example, if we add together a whole bunch of glucose molecules to make a molecule of starch, a polysaccharide, where all of those glucoses are alpha anomers, then we have starch. And now starch is great, right? We can eat starch. We have enzymes that can digest that starch into the different glucose molecules, and we can get energy from it. However, if all of those glucose molecules that make up this polysaccharide are beta anomers, then instead of having starch, we have cellulose. And cellulose, we don't have the enzymes to break them down. So you can eat, for example, all this cellulose, so like all the plant material and the plant cell walls are all made out of cellulose, and we simply cannot break that cellulose down. That is insoluble fiber. It's helpful to clean us out, but it's not going to provide any energy for us. So, anomers, they look subtly different, but they have very big consequences in the structure and function of the molecules they are involved in. All right, so make sure that you go back and you review all the different ways that two monosaccharides can be different from one another. And now we go into disaccharides, which is nothing more than two monosaccharides connected to one another. And they connect to form what's called a glycosidic linkage. And the way that works is it's always between the this carbon here. Okay, this carbon here is the carbon that got attacked by the oxygen in forming the ring. Okay, this used to be that carbonyl carbon. Well, that is always going to be covalently bound to one of the oxygens in one of these hydroxyl groups. Okay, that's always going to be the case. It is a dehydration reaction, so we get rid of a molecule of water, and then we end up with this glycosidic bond. Now, there's some very common disaccharides that you might be familiar with. Two glucose molecules give you maltose. That's the disaccharide, that's the sugar that gives that unique flavor to a Whopper, those malt balls. Glucose and fructose give you sucrose, that's table sugar. And glucose and galactose, that makes up lactose, that's the milk sugar, right? Some of you may, or many of you in fact, are probably lactose intolerant. You do not produce the enzyme anymore that digests lactose. It instead gets digested by bacteria within your gut, producing gas, and it can be uncomfortable. Okay, polysaccharides now are sort of like disaccharides taken to the extreme. It's adding those monomers, adding those monosaccharides over and over and over again. So you've got hundreds or thousands of monosaccharides that are bound together. Sometimes they can be the same monosaccharide over and over and over again. Sometimes they can be different monosaccharides. They can be linear or they can be branched. Now, we don't have it on this image here, but this is a linear chain. What might happen is that we might get another chain going off one of the hydroxyl groups in another direction. We'll see an example of that in a moment. And they could be made up of alpha anomers or beta anomers of the monomers of the monosaccharides. All right, let's take a look at storage polysaccharides. That's our starch and our glycogen. Now, these storage polysaccharides, they're always chains of glucose molecules. Those glucose molecules are always in their alpha anomeric state. Okay? In plants, these polysaccharides are starch. So here we have starch, and we have two types. We've got amylose, we've got amylopectin. Notice amylopectin has this branching here. The starch that you find is here in the endosperm of a wheat, a grain of wheat. So we have the bran, that's going to be the protective layer. We've got the germ, there's the little baby plant. And then we have the endosperm that is primarily starch. And what it is, it's a fuel source for this growing little baby before it develops the ability to have photosynthesis, to perform photosynthesis. But what we do, right, is we grab all these wheat kernels, we get rid of that germ, we get rid of that bran, and we grind up that endosperm, and lo and behold, we've got flour. So flour is pretty much 100% starch. Now, glycogen is how animals store glucose molecules. And when you take a look at glycogen that are stored oftentimes or within granules within the cell, they look like this. They look actually much more similar to amylopectin than amylose. It's got a lot of branching. Now, that is in contrast to structural polysaccharides. Now, structural polysaccharides still consist mainly of glucose monosaccharides connected over and over and over again. But the difference here, and this is key, is that they are the beta anomers of glucose. And because of this slightly different structure, what happens is the different chains of polysaccharides, because they are in the beta anomer, they have the ability to form hydrogen bonding with each other very tight. So they form these fibrils. And that means that they're insoluble in water, right? If you take starch and you slap it into water, it's going to dissolve. If you take cellulose and you put it into water, you're not going to get it to dissolve. Cellulose is the main component of cell walls of plants. So if you take a look at hay, for example, out there, a bale of hay here, it's pretty much 100% cellulose. Your t-shirt, pretty much 100% cotton cellulose. Chitin is another type of structural polysaccharide that's responsible for the integrity of the exoskeleton of insects and fungal cell walls. And both of these involve monomers, so glucose monomers that are in the beta anomer. Okay, and that's why they have these unique properties. They won't dissolve in water and they're very tough. So again, make sure you go through this video several times, take notes, be ready to ask questions, and be able to do each one of these learning objectives.